A very warm welcome to everyone. I am Abhinav Baranwal and I am going to present on the topic mechanism of generation and propagation of nerve impulse. Now before I dive into the topic, first of all we will discuss about neuron, its structures and functions. So a neuron is a special kind of cell which carries signals and is also known as the functional unit of nervous system. A neuron has nerve cell and its all branchings. It is formed from neuroblast and it's the longest cell of the body. A neuron consists of cell body, axon, and dendrites. A cell body has nasal granules, which has a number of small basophilic granules formed of rough endoplasmic reticulum with ribosomes and are site of protein synthesis. Cell body contains neuroplasm, which is essential for high energy impulse conduction, as it has large number of mitochondria, so it provides more energy, which helps in conduction of impulse. Now, interesting fact about neuron is that it lacks centrioles. It has no centrioles, and we all know that centrioles helps in formation of spindle fibers, which in turn helps in cell division. So a neuron cannot divide. Okay, a cell body has extensions, as you can see in the image. These are called dendrites. Dendrites are extensions away from the cell body. Dendrites helps in receiving messages at the point of synapse. Synapse is the point of interaction between two neurons. So main function of dendrite is to conduct nerve impulse towards the body. And not all neurons have dendrites. Like unipolar neurons, they lack dendrites. Now, the axon body has a sheet-like covering called myelin sheaths. These enclose the axons and provide insulation. These are very helpful in transmission of signals. Moving on to the topic, generation and propagation of nerve impulse. So, nerve impulse. Nerve function depends on charge difference across a neuron membrane. Outside it is positive and inside it is negative. That is the resting condition. So at rest, a neuron's plasma membrane has potential energy, the membrane potential, in which just inside the cell is slightly negative and outside is slightly positive. The resting potential is the voltage across the plasma membrane of resting neuron. Now, as you can see here in the image, outside of neuron there is more sodium ions, and inside is less sodium ions, whereas inside there is more potassium ions and outside there is less potassium ions. Now this causes resting potential. Now because of this there is outside more positive and inside more negative. Now the resting membrane potential, also known as polarized state. In resting nerve fiber, the cytoplasm beneath the membrane is more electronegative, whereas outside it is more electropositive. Now resting membrane potential is established at minus 70 millivolt, negative minus 70 millivolt. Okay, so one thing about resting membrane potential is that it has poor permeability for sodium ion, whereas higher permeability for potassium ion. Therefore, potassium can easily cross and sodium have difficulty in crossing. Now, how this gradient is maintained, how the polarized state is maintained? It is maintained because of sodium-potassium ATPase channel. It is an ungated channel where there is efflux, that is outside movement of every 3 sodium ion for every 2 potassium influx, that is inside movement of potassium ions. Moving on, so in resting membrane potential we have seen that it is fixed at minus 70 millivolt, which symbolizes or states that it is ready to receive a signal. So whenever a signal or stimulus comes from brain or spinal cord and the neuron hits it, receives it, there is a rapid localized temporary inflow of sodium ion. Now, so as you can see in the image, sodium moves inside at a particular region. Okay, that region is called triggered zone. Now sodium moves in. Why it moves in? Because of a difference in concentration gradient. So what happens is outside there is high concentration and inside there is low concentration, so sodium moves inside. Okay, now see in this image, there is outside membrane is positive, inside is negative. Here in this region, the trigger region, when sodium moves inside, the concentration gradient from minus 70 millivolt changes to plus 20 or plus 30. Now this change is called depolarization. Okay, now this is in one region. Now what will happen, this will move in a waveform till the end of neuron. This movement till the end of neuron is called action potential. Okay, how it moves now, see, we know that one region is depolarized. What will happen, there will be a series of depolarization till the end. How it happens, you can see here in the image that this region is currently depolarized, and now the region next to it will have a difference in potential gradient. So this will produce a small local current in the area, and the local current will become a stimulus which will cause the sodium channel of this region to open. Now sodium channel in this region will open, again sodium will move in, this region will depolarize. This will again create a difference in potential gradient and will act as local current or stimulus for the next region. Same thing will happen there. This will keep on moving till it reaches the axon end. This will keep happening till it reaches the axon end. Now you might be wondering, if this is happening, so all sodium channels will open. No, it won't happen like that. What will happen in the prior region will be called as repolarization. Now see, there is potassium opening. Okay, so in repolarization, what happens, when a sodium channel closes, it initiates the opening of potassium channel in that region. So what will happen, when a sodium channel moves forward, the prior channel of sodium will close and in that region potassium channel will open. Now what happens when potassium channel opens excuse me, when potassium channel opens, there is outflow of potassium ions. Now from excited stage it comes to the resting stage again. Now this is called repolarization. But one interesting thing about potassium channel is that these channels are very easily opened, but they do not close very easily. So the resting membrane potential should stop at minus 70 millivolt. But what actually happens is it goes further. It goes till minus 80 or minus 90 millivolt. Now this is called hyperpolarization. So the channel closes at minus 90, but till then it is hyperpolarized. Now from hyperpolarization to resting stage, this time period you can see from the graph, this time period from here to here is called refractory period. Now refractory period is a time period when there is no stimulus, when no new stimulus can be received until it reaches the resting stage. Now you might be thinking, if there is no stimulus and say I put my hand on hot iron, I will not feel it burn. But one thing about refractory period is that it is so small. It's almost like 1 over 2500, or you can say 4 into 10 power minus 4 second. It is so small that it does not matter at all. Now if you will look at the graph here, you will see a failed initiation. Now failed initiation depicts all or none phenomena. What is all or none phenomena? All or none phenomena is that when a stimulus is received, it has to be at least that much powerful to reach the threshold, so depolarization can occur and action potential can form. If the threshold is not that much powerful, it will not be regarded at all or none. Either it has to be 100% or it has to be 0%. Nothing matters in between. So that's how propagation of nerve impulse works. Now let me summarize what I just told you. Resting stage, in resting stage, voltage gated sodium and potassium channels are closed. Resting potential is maintained by ungated channels. Then next, a stimulus opens some sodium channels. If threshold is reached, that is all or none phenomena is true, then an action potential is triggered. Addition to sodium channels open, potassium channels are closed. Interior of cell becomes more positive. Now sodium channel closes and inactivates. Now potassium channels open and potassium rushes out. Interior of cell is more negative than outside. Now the potassium channel closes relatively slowly, causing a brief undershoot or hyperpolarization. Then there will be a refractory period. Then it will return to resting stage and be eligible for new impulse. There are some references, and thank you. Questions?